Cameron McIntyre, works for the Forest Service.
Perfect. All right.
He's the guy I think about when I think about beech leaf disease.
It's not my fault. Okay, let me just make sure we're streaming. Yep, cool. Hey, good morning. Thanks for coming to the Forest Health Seminar. I think this is the third consecutive year I've been asked to talk about this topic at this meeting, which is okay. It's an emerging disease. There's new research coming out all the time, so I'm always trying to infuse like the latest information out there. But I'll start just by giving like a little bit of a primer on the biology of this disease for folks who are unfamiliar, and then I'll talk about more about what's new and what's being done out there. Okay. Oh, perfect. So host of beech leaf disease, we only have one native Fagus genus in North America. That's our American beech, Fagus grandifolia. There are 3 other hosts that are susceptible to this disease that are worth mentioning because they're very important to our nursery industry. They're commonly planted in urban settings. So European beech and all of its cultivars are susceptible to beech leaf disease. This is also really concerning for our friends over in Europe because European beech has a much more important economic hold on their market there than it does in North America. And then it also affects some of the Asian beech species, Oriental and Chinese beech, which are also quite popular cultivars. So we think we know what's causing this. It's a nematode. It's a parasitic foliar nematode called Letolincus crenate, subspecies McCannii. Nematodes are cryptic little animals. They're worm-like, but they're not closely related to worms that you think of like earthworms. They're dimorphic, so they have male and females, but the population that we see here in North America is predominantly female because they're parthenogenic, so they can reproduce asexually. It doesn't take a lot of females to initiate an infection, we don't think. The video that you see here is sort of like what you might expect to see when doing diagnostics for this disease. It's quite easy. If you have some material that you suspect is infected, you can just cut it up, you can put it in some water, and the adult nematodes will just preferentially start to swim out into that solution. This doesn't happen all times of the year. The adults are typically active starting in October and September. So there's a couple like qualitative symptoms that I like to group into some categories here. What we see with these light symptoms is typical for new and initial infections. We often refer to this as banding. It can create these either light or more often dark colored bands or galls within the interveinal secondary veins on the leaf. The number and the size of these bands can be really variable. And you can see from that photo on the right there, you can actually detect these falling senescence. So you could potentially do some winter diagnostics by looking at leaf litter. And then we approach like these more heavy symptoms as the disease progresses. This is often referred to as crinkled leaves. They're curled and shriveled along the edges of the leaf. The morphology is also tends to be stunted, so they're smaller leaves in general. And they have this like thick, leathery feel as well if you were to pick some up. And as this progresses, it's also quite common to start seeing things like yellowing, so a chlorosis that progresses into necrosis and tissue death. I also like to highlight some of the look-alike symptoms out there. There's a lot of things that kind of look like beech leaf disease that we see and we get a lot of pulse positives for. Probably the most common one is consistently the woolly beech aphid. It causes some of like that curling and coupling of the leaf. But if you were to, turn these leaves over in the summer, more often than not, you'll find a bunch of white aphids on the bottom that can be diagnostic for that. The other native insect pest that we see on beech is beech earinium mite. This doesn't cause a lot of damage, but it can proliferate the galls that you see in that center photo. And then last year, we've already talked about frost damage. Beech is quite susceptible to the late season frost because phenologically it is one of our last hardwood species to start bud break. And so we saw a lot of damage that kind of looked like beech leaf disease, but once we took a closer look, we were usually able to diagnose that as frost damage. So the progression of the disease is pretty interesting, or really the lack thereof. Like most of the foliar diseases that we work with are fungal in nature, and so they have like this defined like growth and development period. They have to like produce hyphae and fruiting bodies and spores, but that's not the case with beech leaf disease. All of the damage that's done is actually done on those primordial developing leaves while they're still inside the buds. So a leaf that pops out in the spring as asymptomatic isn't going to develop symptoms necessarily later in the year. Generally, the same is true for banded leaves. If a leaf comes out with 4 bands on it, you're not going to see 5, 6, 7, 8 bands develop later in the year. With crinkled leaves, I will say there's a little nuance here because we can actually experience defoliation as that necrosis advances. But the one symptom I haven't talked about is this bud abortion. So if the nematode populations overwintering inside the buds reach this critical threshold, the damage done to the primordial leaves is so severe that they either just fail to leaf out or the buds can fall off the stem completely. And this can lead to 2 different conditions. One is this non-foliated state, which looks like a defoliated stand, very thin crowns. But there can also be some amount of carbon allocated to producing new buds and new leaves, so you could expect to see a second flush. But by and large, what we see, those second flush leaves in no way compensate for the total amount of foliage that we're seeing lost by this disease. So this is stepping back. This is looking at what some of those impacts you might expect to see more on the macro level. Very thin crowns resulting from multiple years of infection from this disease. And this is more of a shot of the understory changes you could expect to see. So not surprising to experience large changes in the understory light environment. This has implications for regeneration in stands like this. This is from a site in southern New Hampshire. This is at Bear Brook State Park. This is one of our first infections we found in the state in a natural stand. And I think to date it's probably the most severe that we have. That photo on the left, I believe, is 2 years post-infection. And you can see that compared to another beech-dominated healthy stand. I have a little bit of data that I wanted to share at this meeting that I've collected in these stands. No one had really looked at like bud retention and leaf out following this disease, and so I did some repeated measures across a couple stands in this area. And so the first thing I'll show you is bud retention on this graph here. That control plot is shown on in the black circles there. Our response variable here is the number of buds standardized by branch length. And what we can see straight away is we're actually looking at a couple different cohorts of buds here. So I did some measurements in February and April last year, so that would have been the previous year's bud cohort, and then a measurement in September, which would be the buds that are currently on the trees, the ones that we're going to see leafing out this coming spring. And so what we can see after 2 years of infection, we see a relative reduction of about 24% between a diseased and a healthy stand. And as we approach our 3rd year of infection, it looks like that's increased even more to the order of about 42%. That photo on the top left there, I took a photo of all the branches that I measured. It was over 100 branches. You can see I've just highlighted with those red circles like areas on an infected branch where you would expect to find buds, but they're either not there or they just failed to leaf out. And then I also want to highlight, if you look at that photo in the bottom right panel, we actually did find some banding in what we thought was our healthy stand. I mean, it was healthy in the previous year. So now we're in a position where we can track sort of what that initial infection looks like. In these stands, I also measured leaf out and the symptoms that we observed at leafed out. So in that control stand, you can just see like the sheer number of leaves is much higher compared to the disease stand. This was quantified at 2 different times, so looking at June just after leaf out and then September. You can see in September what's coded in like that dark green color, those are second flush leaves. So you can see that these trees are making an effort at a carbon expense to try to recoup some of that lost foliage. But as I mentioned earlier, it seems like there's no way that it's going to account for that total amount that's been lost. I guess I will point out too, you do see there is a little bit of second flush in the control plot, but we attribute that to the frost damage that year. So we do measure nematodes in all of the buds that I looked at in that Bear Brook study. I'm not going to break down the data. I just wanted to like show you what we're finding like in individual buds. It's a ton of nematodes. This is a video, so you can see things squirming around. Nematodes have the propensity to like aggregate into like those cotton ball-like structures. They call it nemowool. I mean, there's hundreds of them in those little balls. It's really hard to overstate just like how many nematodes are in infected stands. This is from a single bud. We're seeing on the order of like 6,000 to 9,000 nematodes. So if you even try to do some like allometry to scale that up at how many nematodes you might expect to see at like the tree or the stand level, I mean, there's too many. there's millions of them, right? And it doesn't take much to initiate an infection. And I think that's part of the reason why we're seeing this disease spread so fast. I do want to highlight some recent work that's been done by my colleague Paulo Vieira. He's a nematologist with the USDA Agricultural Research Station. He's the person that figured out that we can actually detect symptoms on the buds. So you saw Mike from the state of Maine talk about how, there's applied ways to use this information. They're able to do some bud surveys in the wintertime, which I think is really cool. I'm glad that they're doing that effort. This also translates to that leaf development. So we see, as we see this proportional increase in damage to the buds, we see that correlating to the damage that we see in the leaves. And this is also a function of the number of nematodes that we find inside the buds. This is my favorite photo from Paulo's paper. This is actually the cross-section of a bud, which I think is really cool. I had never looked at this before, so I put some labels on here just to like orient us to what we're looking at. So that like folded up accordion structure that you see in there, that's a single young developing leaf, and then like that circle in the middle would be like the primary vein, the midrib. But what I want to draw your attention to here is the secondary veins. So if you look at like the peaks of those folded points, those are the secondary veins. So those are the ones that come off of the midrib, and those are where we typically find the symptoms of beech leaf disease. And if we look at where we are finding nematodes in these cross sections, that's where they're hanging out. They're just kind of housed like inside of those little ripples. And so I think this is probably some of the best evidence that we have of attributing the nematode as the causal agent for this disease. It's clear that this disease does not exist without the nematode, but I also have to recognize that there is a lot of ongoing research out there by several lab groups looking at either fungal or bacterial symbionts that may be associated or possibly vectored by the nematodes, because we're not actually sure like the mechanism of the damage, like whether or not it's direct feeding by the nematodes, if they're carrying something with it, if they're secreting some kind of proteins. We haven't quite unlocked that piece of the puzzle yet. Okay, I'm going to pivot now to big picture looking at spread. You've already seen spread maps. I'm showing you an outdated one right here because there's been some really interesting developments, I guess you could say, with the way this is moving around. It's like not a very obvious advancing front. You can see from that initial infection point just south of Lake Erie that spread was actually really slow for like the first 6 or 7 years that we detected this disease. And then we had it pop up in New England and southeastern New York. We know that that part of New York is a very common hotspot where we see lots of pests and diseases get established, so that's maybe not so surprising. But I like to just kind of flip through so we can really see how this has developed over the last few years. So this is current through 2020. This is 2021. That bar graph is showing the cumulative number of county detections over the last few years. So in 2021, we can see like that corridor between these two satellite infestations seem to have connected. And then we had another satellite infestation pop up in 2021, which was that damage up in Maine, which seemed to be sort of an independent event potentially. And what was really interesting about that Maine infection was it wasn't just like a few trees, it was everywhere and it was severe. By 2022, I think that was actually the most individual detections that we had in a given year. That's when we found it in New Hampshire. And then this is the version that you've already seen. We had a lot of detections just all along the margins of this disease front. And as Josh mentioned, that's when Vermont came online as well. So transmission is like really probably our primary like unanswered question with this disease. The Forest Service is involved with some international research projects looking at how this is moving around or whether it's vectored by something. I don't have a lot of data to share on that, but what I can tell you is we find it literally everywhere. Like everything that we look at has nematodes. Like we can look at arthropods, mites. It seems to be associated with abiotic transmission through wind and rainfall as well. One thing I was reminded of recently when I was at the planner yesterday is like we even find it like nematodes just like hanging out like in spider webs in the forest, which tells me that the nematodes are they're just everywhere. They're quickly becoming just part of our environment. I'm going to talk a little bit about our long-term monitoring plot network. So I help coordinate this with my counterpart, Danny Martin. She's the forest pathologist with our Morgantown field office. This covers all of Region 9, but I'm going to just talk about us here in New England. So the black points on this map show all of our monitoring plots that, as of last year, were positive for beech leaf disease. We have a total of 82 active plots right now between New England and New York. We remeasure these annually. Last year, 54 of those plots, or about 66%, were positive for beech leaf disease. And we're now at a point where we actually have over 3 years of data in 68 of our plots. So this is great. What I'm going to share with you today is some of the trends that we're seeing over this 3-year period. So this is just a quick snapshot of what was measured last year. Within these plots, they're not big plots, they're 1/10 of an acre. But we measured 1,600 mature trees. Of those, 700 of them were beech, so about 43% beech by basal area. These aren't like FIA plots, they're not randomly located. We are like targeting areas where there are beech so we can measure the response variables that we're interested in. Of those, beech accounts for about 30% of our codominant trees, but not surprising, it does account for a much higher proportion of trees in that intermediate and suppressed strata. This, of course, would be because beech is probably among our most shade-tolerant hardwood species, so it does do quite well under shade. In terms of some of the impact metrics that we're looking at, the primary ones would just be our ocular measurements of dieback and density that we're seeing in the forest. Dieback is a pretty straightforward measurement of just how many dead branches that we can observe on the tree. Density is more a metric of how much foliage we're looking at. I always struggle to define density, so it's just easier to show you a photo of what that looks like. For example, this photo on the left here would be a tree that we would categorize as having only 1 to 10% crown density. We group things in these bins to make it a little bit easier for our technicians that measure this data. Whereas a tree that has a relatively full crown is something that you would categorize as having, upwards of 75 to 100% crown density. And these two things are related, so there's an inverse relationship between crown density as a function of crown dieback. What this figure is showing in red are all of our plots that were positive for beech leaf disease last year. You can see that there's a lot of points that fall off that line, and that's because there tends to be a lag, right? So typically, declines in crown density precede dieback by at least a year or two. Okay, very similar figure. This time our response variable is the severity of beech leaf disease that we see across our plots. The metric I'm using here is an index of the total amount of symptoms that we see in the crown, and our response variable is crown density. And we do see that there's a proportional decline in crown density as severity increases. Fairly high correlation. It seems like this is explaining at least 50% of the variation in the data. I also did try to convey some of the dieback information here. So the size of the dot that you're looking at on this graph corresponds to higher levels of crown dieback, so dead branches. Let me talk a little bit about the changes that we're seeing over time. So what I've done here is I've grouped all of our sites into the number of years that they've experienced infection. So I couldn't just show you like consecutive years of data from 21, 22, 23, because the timing of infection like changes all across our plot network. So I've just grouped them in terms of the duration that they've been infected. And what this graph is showing is actually the proportion of the healthy leaves that we observe in the crown. This is approximately linear. We see a steady decline in healthy leaves. By year 3, it seems like maybe around 20% of our crown is healthy. Similarly, looking at banded leaves, we would see the inverse, like we would expect. So there's a proportional increase in the amount of symptoms over time, and that increase is even more pronounced in these advanced symptoms. Typically, like the light symptoms are really only common, I think, in like the first couple years of infection, but the progression can change very dramatically. Some sites, we see a very slow progression of symptoms, and then other sites, it's sort of just like flipping a switch, where you go from a first-year infection, you have a lot of crinkled leaves, and then the following year you have just widespread bud abortion. We're not really sure what causes that amount of, that severity level to accelerate at that rate. These are the impact metrics I mentioned earlier, so looking at relative declines in crown density between year 1 and 3. It's on the order of about 44%, so we're losing almost half of our crown density following 3 years of infection. And then inversely, over a 2-fold increase in the amount of dieback that we're seeing. This is probably a good point to mention that in our monitoring plots, we haven't seen a lot of mortality yet. Like, we know it's occurring on the landscape, but it's really patchy. It's probably you know, a matter of time. I don't really like to make predictions about it. But one thing I do like to talk about is we can't study beech leaf disease in a vacuum, right? We know we have this legacy of beech bark disease. There are multiple things that are affecting the health of our trees. And we do try to quantify some of the metrics of beech bark disease within our monitoring plot stands. So we have this legacy of beech bark disease in New England for over 100 years now. It's spread relatively slowly from Nova Scotia, and it's only just reached parts of Ohio in the last decade or so. But you know, if we try to generalize the advancing front of beech leaf disease, they're kind of moving in opposite directions, but within the last 3 years or so, we've really reached this intersection or this coalescence of the two diseases. And so I'm really interested in using some of this monitoring plot data to try to tease out, I have a minute? Sorry, guys.
2 minutes.
So the short of it is we don't see any correlation between the amount of cankering on a tree and the amount of symptoms that we find. But what we do see is the outcomes of having the combination of these two diseases is much worse. So what this figure is showing here is the relative crown density between different disease combinations, okay? So what you're seeing in blue are trees that don't have any disease. These would probably have some kind of resistance to the bark disease, presumably. And then what's shown in orange are trees that have a combination of the two diseases. And what we see there is a relative decline of about 27% with respect to the healthy trees in this case. This doesn't account for the number of years in infection. This is actually just looking at a snapshot in time. If I were to pull out those 3 years of infection, this would be exacerbated. One thing I did is you can break this out by relative severity within these classes. We start to see declines on the order of about 40%. So if you wanted to start making predictions about mortality, these are probably the trees that I would keep an eye on. And my last slide is just talking about treatment. There's not a ton of options out there right now. The one you'll see marketed by arborists is actually this potassium fertilizer. It contains a phosphite molecule that seems to be an inhibitor for nematodes. It actually does cause some symptom recovery and lower number of nematode densities in the buds. So this is promising stuff. So far it's really only been piloted in urban settings, but I'm going to be working with folks at the Hubbard Brook Experimental Forest this year to try to scale this up to landscape scale treatments. I don't think it's that crazy of an idea. The application is actually very similar to what people use on hemlock, using imidacloprid to treat things like scale and adelgid. It's just 2 ounces of chemical that you scale for every inch in DABH, and you do a root drench. So we're going to play around with dosage response. It's really onerous to expect people to apply something like this twice a year. So maybe we could double the dosage and get away with a once a year application. So maybe next year when I'm asked to talk about this, I can share some results. I'll just thank all the state partners. I don't measure all those monitoring plots. I really rely on folks like Josh and Bill and all the state forest health agencies to help with that work. So thanks for listening.
There's been so many efforts made to control beech.
To like suppress beech, you mean?
suppress
Yeah.
speech. So it's like, I don't know who to root for here. You're like, is that becomes my best friend now, or it's odd situation.
Yeah, that point's well taken. A lot of people are I show this side and like, why would you treat? You know? It really depends on your management objectives, I think. From a timber standpoint, sure, this could be like the next greatest thing. But for the people in the wildlife community, this is bad news. So I get both sides of the story there. I mean, the real thing is like beech is like such an abundant and foundational part of our forest to just like, if we're like looking down the pipe at like a chestnut situation, then that would be terrible for our forests. So I hope that's not the case.
Other questions?
It's been in Ohio for a while. What are the stands that have had this for 10 years look like?
They don't look good. They don't have beech bark disease there, or not much of it though. So I think it's a little bit like looking into our future. My prediction is here in New England, we might expect to see that accelerated. They see a lot more mortality in the understory. It seems like within the last few years, they're seeing a lot more overstory mortality. In places like Connecticut and New York, it's only been for 4 to 5 years. We're already seeing that. So I think we're just in a position where our trees are already pretty stressed on top of this new disease.
Awesome, thank you, Cameron.
Yeah, thanks everyone.